Hey guys, so today we are going to be taking notes about gene expression. Alright, so first we're going to go over what the heck is gene expression. So remember, genes are just a segment or portion of DNA. And when we express a gene, that means that we take a gene and we transcribe and translate it into a protein. Proteins are also called polypeptides. Poly meaning many, and peptide is just the bond that links amino acids together. Okay, so we're first going to review gene expression. So remember, this is essentially the process of DNA turning into protein. That's DNA to mRNA to protein. All right, a little review of what we've gone over in the past. Genes are DNA, just a tiny portion of it. So like, if I have DNA, a little portion of it would be a gene. DNA is made of nucleotides. It is just a sequence of nucleotides, and a nucleotide has three parts: a phosphate, a sugar, that's a little house, and my nitrogenous base. Okay, my nitrogenous base. Remember, in DNA, I have 4 nitrogenous bases. That's A, T, G, and C. Only in DNA. All right, so we're going to do an example. We're first going to review how do I get from DNA to protein. This is called protein synthesis or gene expression. So this is protein synthesis, and this is how genes are expressed. It's just when they turn into protein. Okay, so let's do a little example. So when I'm writing a gene, remember, I'm just basically going to write my sequence of nucleotides, which I do just by the nitrogen bases. So let's say I have a gene that has A, T, G, C, G, A. Remember, every letter equals a nitrogenous base. Alright, so that was my DNA. Next, I'm going to transcribe it. So transcription is when I go from DNA to mRNA. So this is transcription. And when I transcribe, I'm using the base pairs A to U and G to C. So A, because RNA, remember, has no thymine, it only has uracil. So adenine to uracil, thymine to adenine, guanine to cytosine, cytosine to guanine, guanine to cytosine, adenine to uracil. All right, now once I have my mRNA strand, I now need to divide into codons. Remember, codons are every 3 nitrogen bases, and then I use this very handy chart to my right in order to translate mRNA to protein or my polypeptide. So mRNA to protein is translation. That is mRNA to protein. Transcription, remember, it was DNA to mRNA. Here we go. All right, remember my fancy codon chart. So I only look at my mRNA strand because look at the title of my chart, RNA. Don't look at DNA for this. Okay, my first codon is UAC. U, I start on my left. It has to be in this row. A needs to be in this box. C needs to be in this row. So this is tyrosine. Every 3 letters represents 1 amino acid. GCU, G needs to be in this row. C needs to be in this box. U needs to be in this row. That means this is alanine. So these are my chain of amino acids. Okay, now what the heck does this have to do with mutation and gene expression? So let's say that I mutate my gene. Mutated gene. All right. So let's say that I now have a gene that is A, T. Now I'm going to change a single base, which means that this is a point mutation. GA. This is my DNA. I'm going to convert it to mRNA. Okay, still divide it into codons. So we see right away that my DNA sequence was different, now my mRNA sequence is different, and now my amino acid sequence is going to be different as well. So let's look at this. Okay, so if I have U to A, that's my new codon, I now create a stop codon. Stop. If I have created a stop codon, that means a nonsense mutation has occurred. Okay, this is my protein. So my protein really doesn't even get made. So if there is a change in DNA, which is a mutation, then there is going to be a change in the protein or the gene that's expressed. Now I want you to imagine, remember that there were two processes that we talked about, mitosis and meiosis. So if this were to happen in mitosis, that means that I'm dividing regular cells. So if I'm changing in mitosis, remember I have like 37 trillion cells in my body. So sometimes when we change even one letter, like if this protein doesn't get made, right, maybe then we have stopped the control of the cell cycle and now my cells are dividing uncontrollably and I get cancer. And because my cells are continuously dividing, maybe like this prevented a checkpoint in it, then perhaps my cancer could grow. Let's say that this mutation happens in meiosis. So if, oh, sorry, this is a lot of writing. So if mutation, this is another bullet point. If mutation occurs in meiosis, remember meiosis produces your gametes or your sex cells. So if it occurs in meiosis, then that mutation, if that's specifically like the egg or sperm that's used to make a child, the mutation will be carried to your offspring. Offspring is like a child. So let's say that there's a mistake in meiosis. There's a mutation in the sequence when meiosis is happening, and let's say that that lucky egg or lucky sperm is the one to come together to form a child, then that child will now have that mutation, whatever it is. So there are some mutations that are like, okay, that's fine. Sometimes it doesn't even change anything. They're called silent mutations, but then in other times these mutations can be like extremely detrimental. Like it can cause things, maybe like hemophilia, which is when your blood can't clot and like you get a paper cut and bleed to death, you know? So it can just have an effect. The main effect though is because I'm changing my protein. Whenever I change my sequence, then my amino acid sequence changes, and then my protein shape will change, and then function always follows form, which means that if I am changing my shape, then my function is also going to change. So proteins always fold in on each other, but if I'm changing the shape of my protein, then I'm also changing what it does, and that could be really, really, really bad, really consequential. All right, that was it. Sorry that was long. It was very interesting. Okay, have fun.